Hey, what's up, everybody? Welcome to the Magic Mushroom Speaks podcast. I'm your host, Traveling Psychonaut. On this podcast, we have a guy who really knows his stuff and is well known around the Discord communities for all his great work, his YouTube videos, and his wealth of knowledge. For this episode, I'd like to introduce to you Edward. Sorry, guys, Edward Grand to the show. I'd also like to take a minute and shout out Simply Mycology. Our mission is simple: supply products to the home mycologist at the lowest price while maintaining the highest quality. Unlock your mycology potential with a handcrafted small run grain spawns, substrate, and agar. No more worries about quality control issues that plague large scale operations. Our perfectly hydrated substrate, fresh agar, and grain spawn bags are tailored exactly to your expectations, just the way you would do it, so you can put all your focus into growing your dreams with confidence. Now, I'd like to welcome Edward Grand to the show.
Hello.
Hey, how's it going, man?
Hey, it's going pretty good. Oh man, that works like seamlessly. I was really a little bit worried. So you can hear me
Yeah,
okay?
yeah, I can hear you good.
Ah,
Hey,
sweet. Okay.
I'd just like to say, I'd like to say hi to my wife. She's listening right now.
Oh, okay, good. My girlfriend's sitting in the back too.
Yeah, see, she's right there.
I told her to keep quiet already though. So.
So how's your day going?
Oh, pretty good. Yeah, it went pretty good. It's already like night here. It's like 10 p.m. here.
Oh man, that's
So
crazy.
it's like the time thing. I saw people asking on the Discord, like, "Oh, where are you guys?" And I was like, am I late?" So I was like, "I think I'm okay."
You can't be
Yeah,
late for your own. You can't be late for your own show.
yeah, I guess so. Well, I'm kind of like kind of like OCD about that kind of stuff. I freak out about schedules and things like that. But now that we're here, it's all good. My phone's charged up and everything. I got the power bank ready to go if something happens.
So you're in for the long haul then.
Yeah, well, also I gotta say, Samsung. I don't have an iPhone like a lot of people. On a lot of the other podcasts, I've heard people like, "Oh, my phone battery's dying." It's like, yeah, that's Apple.
All right.
That's it. You need to buy a new iPhone because like the battery, you can't you can't replace any of the batteries now, right? But yeah, I was just chilling at home. I just saw a new Hamilton Morris podcast. Man, he interviews some really crazy. He had some guy on there like talking about his old school like 1980 like synthesis, and like wow. Um yeah, that Hamilton Morris guy, he's an interesting character, but he's got a lot of good stuff and a lot of thought-provoking stuff about the chemicals that some of us might ingest from time to time.
Yeah, I ingested a little bit last night, so
Oh.
so
Yeah.
yeah, well, let's get on to the question here. Why
Yeah,
don't you
man.
tell everybody how you got started doing the mycology? What put you on that path and that career?
Okay, yeah, if you want to call it a career, I guess it's more like a hobby.
Or a hobby?
Maybe
Yeah, okay, we'll call it a
I'm not
hobby.
really sure. It depends on the day and who I'm talking to. It might be a hobby or a career. Job sometimes. At 4 in the morning and you're standing there like, wow, I'm like making substrate at 4 a.m. like that. This is more like a job. but,
It feels like that
but yeah,
sometimes, eh?
sometimes yeah, you just gotta push through, man, and be like, all right, it'll be better. It'll be better soon. But yeah, I grew up just basically foraging for like wild edibles, and I kind of grew up in the woods essentially. Man, my I'm from Michigan originally, and it was like it's a pretty, I don't know, like outdoor centered kind of culture for most people there. I mean, I grew up like making tree, for what do you call those, like what do we call them, huts in the in the forest, little lean to
Tree
big,
forts,
big
tree forts, or something.
tree forts. Yeah, that's it. Sorry, my English is pretty bad, dude. I've lived
That's
here
all
for
right,
a
man.
long, and my English sometimes I forget words. But yeah, I just grew up like doing the same stuff, making like BMX tracks in the woods, and looking for. So eventually, I started finding all these like. I noticed all these like mushrooms, like chicken of the woods and hen of the woods and shaggy manes, and I was like, man, I bet you could eat some of these. And so I this was way like at the very, very sort of beginning of the internet, and I just happened to like see some website that was for the Michigan Mushroom Hunters Club, and I hooked up with some of they were, I shouldn't say old because they were about my age now, but yeah, I ended up like contacting a few of those people, and I started going on these. They call them forays, like where you basically go out and you hunt for mushrooms. And these guys got me turned on. Well, these guys and girls. I'm from Michigan. When I say these guys, I mean men and women. That's like the way we say you guys. So yeah, I just started going out, and these. There was one guy in particular, Larry. He was really into the microscope stuff, and he had all the books, like the old books. I don't know if the people out there know, but there was this Alexander Smith who was like really super important in mycology back in the 60s and 70s, and I think even into the 80s. He published all these books about like, oh, the genus Lactarius in Michigan, and these weird genera. Like he did things on like Inocebi and like Cortinarius, and I found these books in the library because I went to school. I went to school at Michigan, and then like they had an herbarium there, and I got hooked up with this guy. I think it was Robert Schaefer, I think was his name, and he wasn't really well known. He was more like one of those like people that studies mosses and stuff like that, like cryptogams they call them. And yeah, I just got this guy like hooked me up with a microscope and gave me a little table like over in the herbarium they call it. It's like kind of where it's like a plant museum,
Yeah.
where they guide stuff. And yeah, that's long story short. I mean, lots of things in there too, but yeah, basically, it kind of set me on this path, and then I got involved with the website for that club, and I was like the webmaster. Now everybody does web pages, but back then it was like HTML, and like text editing things, and trying to upload JPEGs of that weekend's finds and put names on them, and then I started doing this. I was like the assistant for again Robert Schaefer. He did this sort of ID course through the local botanical gardens. So man, yeah, I was bringing back all kinds of memories. I haven't really talked about this in a long time, and so I would go out and we would have. I think there'd be about 25, 30 students, and we would just go out and look for mushrooms, and then come back and put like Telluride. You know what they do out in Telluride and that kind of stuff.
No,
In
I don't.
Colorado. Well, they'll have like say a couple hundred. Now they charge, I think it's $500 to attend this like 3 day like weekend. But we just come back. Essentially, you send like hundreds of people out in the woods, and they all go out with their little baskets, like Little Red Riding Hood, and come out with like all these random piles of mushrooms, and you sit there for hours trying to sort out which ones are which, and you put a name on them at least to a genus, like, oh, that's a Lactarius, oh, that's a Coordinarius. You get pretty good at just getting it to genus. And then like what people are trying to do now with the iNaturalist, it was like the original version of what people now do with iNaturalist, where you just go out and now instead of picking the mushrooms, because a lot of places you can't really collect mushrooms anymore, so people will go out and they just take their pictures and they do the whole iNaturalist thing. So this was like the original version of like iNaturalist. Like you'd go there and you'd set them all on a table. We had little cards, like little index cards, those little piece of paper, and you'd label them, and then somebody take some pictures if they were cool stuff. You'd put them in a box, dry them out, and things like that. And yeah, sometimes they ended back in the the herbarium. Sometimes they just ended up in the garbage. Sometimes you ate them if people found good stuff. But it was just like it was like a crazy crazy learning experience, and I got really really good at identifying mushrooms. And I went to my normal engine. I saw, yeah, everybody knows. I was a chemical engineer, so That was like what I was doing as a career at that point, but I just realized it was going to be a very boring career, and I went to work in a lab, so I worked in a medical lab for a while, like doing all kinds of crazy experiments, and I learned a bunch of stuff about DNA and RNA and sequencing and all that kind of stuff. And then I got bored of that, and I like one day literally was doing an internet search. Like it was Netscape back then. That was the search engine that people used.
Yeah.
Like this, like you're old enough to know Netscape probably. Well,
Yeah.
I put in a little search there for like something like mycology programs or something. I was probably looking for other people to chat with about mushrooms, and I mean that's what's kind of cool about this is I'm literally on the other side of the world, like chatting with you guys about mushrooms. You know, this is quite awesome. Like I'm pretty excited. I'm like a little kid now. Like like I get to talk about mushrooms.
That's okay. That's all right. That's what we're here for.
I know. Well, I love it, man. I love it. That's the thing. I was talking about charging my battery. You know, who I can go on for hours, hours, and hours. with mushrooms. Like, what you're gonna have to probably stop me at some point. But yeah. So I ended up getting hooked up with this guy, and like, man, like 2 days later, he was like, I was down in Tennessee, and then it's like, well, I guess I'm doing this, and 4 years later, now I got a freaking PhD in mushrooms. So it's a little bit weird to be honest. What I studied for my PhD was pretty esoteric. You know shiitakes, right? You know
Yeah.
shiitake mushrooms,
Yeah.
shiitake. Well, that's pretty much the genus I studied. So what I thought at the time and for the last 20 years was going to be pretty irrelevant in life has actually turned out to be pretty fortuitous in that I know a lot about the biological species concept and morphology and DNA sequencing, which again I didn't think would apply to our little realm of the mushroom world, but now it does. So people talk about like crossing and making hybrids, quote unquote hybrids. Like I know all about that because that's like what I did for 4 years, like every day for 4 years, like so. So that's so anyway. Then I ended up living in Thailand. That's where I'm literally sitting right now in Thailand. Just kind of again by an accident. Met somebody at a conference. Was like, "Hey, what are you doing in Thailand? Like, I've never been there." And he was like, "Well, you should come." And I came, and then immediately, like the second day I was here, I was like, "I need to figure out how to come back here." Like, this is
So let's just hop
awesome.
on a plane, right?
Yeah, that's literally, well, I had to finish. That was in the third year of my PhD. So I went back and I finished up as fast as I could, basically. And I ended up that same guy or one of his like colleagues that was working up. They were actually working like they were selling T-shirts at like one of these conferences. It was in I was in Norway in Oslo, and just for when you do research you got to go present it to the rest of the world. And so this guy's like, yeah, well, I got a job, you want to come work for me? And I was like, yes. and and that didn't really work out because of reasons I don't want to get into, but it was just a logistical problem. And so then yeah, I just looked for other work, and it's fairly easy to get work here if you're a native English speaker. Like you can get a job fairly easily. I would more than easily. Like as long as you're not a moron and you can speak English, you're like good to go. Pretty much. Like I know a lot of people that are like borderline morons and they still have jobs. So they're doing they're doing they're doing just fine, and they I don't know, they're not the brightest people in the world, but they still they got a job and a happy life.
Yeah, see?
So so yeah, that's a lot. That's a short version of, I guess, a well, long short version.
All
But
right.
yeah, so this is a great
Very
place.
interesting.
This is a great place. I don't know if I don't know. For people out there, definitely look into it as an option, man. If you're in the states or whatever, I've got a lot of friends back there, and I don't know, man. People are not really happy. Well, most of the time I talk to them, so like, just it's an option out there. There's always options.
All right. Is there a easy and easy and quick method for assessing the viability of fungi mycelium growth in the vitro?
Yeah, I looked at that question and I wanted to ask you about that. So in vitro, you mean like in the
Don't
lab?
ask me. I'm asking the questions here.
Okay. So,
What's
well,
up, man?
I'm wondering who asked the question because in vitro literally means like in glass. So I'm assuming they mean like in like a axenic culture, like a medium or on an agar plate.
Let's
I'm
go with
not
the
sure.
agar. We'll take an agar plate because that's most like a lot of people know about that. And
Yeah, that's what I was thinking. So I have to assume they meant like if you have a culture dish and it's got mycelium growing on it, like how do you know if it's still alive? Does that sound
yeah,
what?
that's yes.
Yeah, well, that's quite straightforward. You just look at the plate from day to day, and if it gets bigger, then it's still alive. That's like,
But sometimes
I
you
don't
can't
know.
see it.
Well,
Sometimes
that's
it's
what I
super
don't.
thin, and
That's why I was kind of confused because I'm not sure. Maybe I'm making that question too simple. Like maybe they had a more complex like question that I'm not understanding. I could see with liquid culture they might you might get a little confused because in liquid culture they may be if they're talking about the viability, like i.e. if it's alive or contaminated. That's a bigger question.
well, let's
Like if
talk about the liquid culture because
Yeah, if you have liquid culture, it's sometimes quite difficult. So the problem with liquid culture is it gets bacteria. That's the main reason. Liquid culture gets contaminated, and if you're not like 100% sterile in your procedure, you can get. I mean, I see horrible. I don't want to really on YouTube, I see people doing videos about liquid culture, and I see the main thing I'll see them wipe like a syringe with a 70% alcohol rag. And they're like, well, before you inject the septum, the ship, the self healing injection port, they're like, before you do that, make sure you wipe the needle with alcohol. I'm like, oh my God, no, please don't do that. Don't do that. Don't do that. No, if you're going to be injecting septa, you want to heat. You want to use some kind of heat, either an induction heater or a flame, depending on the needle's like material, the metal it's made out of.
connection.
But most
There we go.
of them use
You're
yeah,
cutting
most
out.
of them they're the mm,
You
yeah,
were
like I
cutting out
said, you.
there.
Oh yeah, that's right. I might yeah, it's like 11 or 10 here. Yeah, sometimes the internet here is not so great, but hopefully it'll
That's all
straighten
right.
up. But, but yeah, I reckon if you're talking about liquid culture, you have to be really, really spot on with your technique because you won't see a contaminant until you either test it on a plate or if you inject grain, you will then notice that if there is literally a single bacterium in there, you might not even see it at first because it'll until you do like the break and shake as people call it, like until you might hit 30% colonization or even more, and then when you break your grain up, whether you're using jars or bags or whatever, you shake the grain or you break it up, and then that bacteria that might be in like a little corner of the bag or the jar down there where the anaerobic kind of it's more wet. Bacteria love anaerobic conditions, like wet conditions. So if you've got grain and you haven't properly hydrated it and you have liquid water, say in the bottom of your mason jar, your quart jar, or you've got it in the bottle of a bag. I like to use bags now, the polypropylene bags. And if you've got a little bit of contamination in the corner and you break and shake, you're gonna spread that bacteria. So you start out with this nice LC, and you got like what you think is LC or good LC, and then maybe you don't test it on an agar plate, and you go and you shoot it into a grain bag, and then when you break and shake, you'll notice that like it just stalls, and that's a big problem because what you've done is essentially taken that little spot of bacteria and you've spread it throughout the entire bag, and then the bacteria is going to win every time. Like bacteria is going to grow and proliferate way, way, way faster than any mycelium. So yeah, that might be what they're talking about. I mean, as far as just knowing whether a culture is alive, it should just be by the growth rate. they could be inquiring about different growth media too. Like I use rice flour now in my media. I use rice flour and agar and yeast extract, or I just use baker's yeast. So I'll tell you that there's a really simple agar recipe that works great. Is just it's just basically rice flour, which what we would call or what you guys would call brown rice flour. Here they just call it rice flour because like all rice starts out brown. so so like if you don't know, like there's a sequence of events that happens. What I use for my spawn is called patty rice, like PADDY, like a rice patty. And when that gets basically hulled, like kind of like when they take off the chaff for like wheat and stuff like that, you got to take off all the kind of the seed coat and the junk. Well, when you get down to what would be considered the brown rice, it still has what's called the pericarp, the kind of the skin of it on there. And so if you make that into flour, it's called brown rice flour. So BRF cakes, what we used to call PF Tech cakes, that is brown rice flour. And what I use to make spawn is like one step before that. So it's still got the kind of like the shell of the seed on it, like yeah, I don't want to make too many analogies to other plants, but it's kind of like wheat. When you harvest wheat, you've got all this chaff on the outside. If you could get that and use that for spawn, it would work really well. But here they sell every variety. They sell what they call patty rice, and then you can buy brown rice, and then there's what we would know as just simply rice, which is white rice. Which here they actually call it like pretty rice, like it's the rice after you've taken off the germ and the endos. Like basically all you've got left is what's called the endosperm, and that's the starchy part, and that's what humans eat in general. Like if you ever, eat like rice, that's what you're eating. It's called the endosperm. Fungi love it, but they also love all the other stuff. Um, so I don't know. I'm getting distracted again here. Sorry. Like I said, you're gonna have to pull me back if I go on a tangent
I'm
because
just
I'm thinking.
listening. I'm enjoying listening to you talk, man. So
Okay,
you enjoy you enjoy yourself.
okay, because I could go on about like rice and different grains for like 3 hours. So you should talk
Okay.
because I teach botany also, so I'm quite familiar with a lot of like botanical. Like probably most people don't know what the hell the endosperm is, but that's like the white stuff that we eat, and I may use fancy words sometimes. So please forgive me. If you don't understand a word, just stop me and be like, "What the hell? What was that, Ed? What was that word?
No
The fuck
problem.
is endosperm? Like, what is that? What is... Sorry, I'm assuming I can swear.
That's
I
okay.
can. Is it? We're not gonna get like demonetized or anything if I swear
No,
on here.
no, I don't think so. It don't matter.
Oh,
So
oh wait.
I seen I seen one of your videos about the unmodified Martha tent. Why don't
Oh
you
yeah, that,
take us through that a little bit?
yeah, that's a good one. My buddy here, he's just setting one up tonight. Actually, he just sent me some pictures before we went on. It's basically the traditional Martha tent that are. I forgot. Do you guys? Do people know centimeters or inches or what? What do
I
people
know
use?
inches.
Okay, inches.
I know
Okay, I do
inches.
too.
But I
I'm
mean, a
American.
lot of the guys from the states and stuff, I think they what do they do? Metric or what is it?
Inches, I reckon. So
Inches?
the tent basically, your standard. What are they? 56 quart mono tubs. Martha tin I use basically has the same footprint as that. It's about the size of like oh god, like it's about oh my gosh, I'm looking at it. It's in centimeters here. It's basically like about 2 foot deep and maybe like 3.5 feet wide. And so it basically takes up the same floor space as a monotub. But the ones I use have four
Hmm.
shelves. So I run shoe boxes now, but in the same tent, you can also put a monotub if it's not too big. You got to work out the sizes and what's, to be honest, most economical for you. Because the tents I have so I'll have like on the bottom shelf, I'll maybe have two monotubs. Sometimes I run them unmodified. Sometimes I do the micro pour tape with the holes, and sometimes I used to do shotgun fruiting chambers. I've kind of done like every variation that probably is out there for mono tubs, and I'm running in bags now too. And when I've been running a lot of my crosses because I'm just looking for spores, I run basically I take a spawn bag. I use a polypropylene spawn a bag, and I just literally like fruit it right in the bag. Like I don't even mix substrate with it. Because for the F2s and the F3s, when you start going through the generations, you want to basically get spores and go back to an agar plate and get the next T0 or the next multi spore plate, and then you go on to the next generation, and then you do that pretty much ad infinitum, like until you get what you want, or you select for something that's hopefully stable. But as I'm sure as everybody's been reading about, the stability of these supposed cultigens or varieties or crosses or whatever, or you want to, what's it, strains. Yeah, it's quite kind of dubious these days because people are putting out so many crosses and so many new cultigens. It's like, wow, I see a lot of disappointment sometimes when people are getting these things and they don't quite pan out to be what they were advertised as. you know,
I think that happens a lot, man. It happens a
Yeah,
lot.
it seems to be happening like with more and more regularity. It's like, wow, I see like every freaking day people are like, why does this look like this? Or why is it, it was supposed to be albino and it's brown, or vice versa. Like, why do I have another albino Avery or Avery's albino? Like, it's like, yeah, why is my emerald whatever not the right color? Yeah, I see it every day on all the groups. It's like it's a common thing, Jack Frost, that's the one everybody loves, and everybody's kind of like, why does my Jack Frost not look right?
Well, it could
It's like,
be a different pheno, no? Like the same, but just a different phenotype.
yeah, that's a whole big can of worms for this day. I don't know if we can go into that like maybe in a no, whole other like podcast. Maybe not today. But yeah, it has to do with a lot of different things. And yeah, maybe we should stay away from that for now because that's a deep, deep, deep rabbit hole. So
Well,
yeah, back to the
you don't think we could dig our way out of it?
um, we might need the help of 100 other people and it would get probably pretty. It would turn into like some like United Nations and like a debate. We'd have to elect like representatives for each continent.
Yeah, we don't want no wars going on or nothing like that.
Exactly. We'd have to get Kofi Annan or something, one of those dudes, to come moderate for us. I don't even. He's the only dude I remember that was the UN guy. But yeah, the tents are awesome. But that being said, I've got like 30 bags now sitting on my floor. So I've got the unicorn. I've got two sides of they're called Pegasus. The distributor now is Pegasus, right? And I've got like 30 of those sitting on my floor. And I ran bags a few times, but I don't. I like that they're cleaner and they're easier, but there's some drawbacks there too. But it's really it's personal preference, like most things in this hobby. There's not like a right way to do it. There's a lot of wrong ways to do it. But there's not really like one right way to do it, but there's a lot of ways to f it up. There's like hundreds, thousands of ways to fuck it up.
Believe me, I know. I've done it.
I know. Well, that's what doing it all these years. Believe me, when you I yeah, I throw like probably a good like a garbage bag full of stuff like every day. Like it just becomes a process. It's almost like therapeutic when you get like a shoebox, and that's one reason I like running in shoe boxes. It's not like such a heartache when you like throw a shoebox away, but if you're throwing away like a 60-quart mono tub, you're like like 4 or 5 quarts of spawn and all that substrate, and it's like
Yeah.
you're just and sitting there with an empty box with a bunch of holes in it. You're like, what the like, oh, like I can't even use that to store anything now. It's full of holes. Man, I used to paint the bottoms black, that stupid crap that was going around like 5 years ago. Like, you got to spray paint the bottom,
I,
that shit.
I seen something about that.
There's still a video out there that I wish they'd take down, and it always pops up on the YouTube search. Like if you just search for like monotub on YouTube, it's one of the first like three that pops up, and it's a the one of the very first thing the guy does is like mask off the bottom of a monotub and take a black spray paint, and I'm like, God, can you stop doing that, please? Everybody knows about liners now, right?
Yeah, yeah.
I hope, but I'm looking at a monotub right now, literally next to me that has like 3 inches painted black on the bottom with a bunch of holes with micro pore tape. It's like that. Like I can't like. It's also suspicious looking. You're right, right? If your mom walks in and you got this weird, or the neighbors see you out in the backyard like painting the bottom of a tote or monotub, they're gonna be like, "What the hell is he doing?"
What's wrong with that guy?
So yeah, that probably didn't answer anybody's questions, but tents are the way to go. But like you said, I have videos on YouTube about a lot of this stuff. And that was part of the reason I started doing YouTube videos, because people were asking, and I was like, it'll be a lot more efficient if I just make a video about this like one time, and then when people ask, I can just be like, go watch this video. Like it's a lot easier.
What What is your YouTube? How can people find you on there?
It's just search Edward Grand. I think I reckon if you just type in Edward Grand, I can send you the link later, or maybe I could. Oh, I think I sent you a
Yeah, I
request
have
to
it.
try.
It's all over the Discords, man. You can't miss it.
Yeah, that's what I thought. Yeah, so just go to any
Yeah, yeah,
one of
but
those.
just for somebody's listening, they don't, if they're not on the
Oh,
Discord or something, they can find you.
oh, oh, yeah, I forgot that. Like, there's normal people out there that aren't on Discord yet. No, I think like we're talking about. I just got on Discord like maybe 6 or 8 months ago. I didn't even know Discord existed till like literally like 6 months ago, and somebody found me on Facebook or whatever and was like, "Hey, go check out Discord." And I'm like, "What the hell is Discord?" I thought it was some like mixing, like DJ software or something. I'm like,
It kind of sounds like it.
yeah, yeah, like chord, and then the first time I looked for it, I actually spelled it like Chord, and I'm like, where the hell is this thing? And like, oh, no H, because chord, like a music chord. And that's what I assumed it was, some music thing, like it's an like alternative music thing. And I was like, oh no, it's about video games. And I was like, oh, okay. Now I
Yeah,
know
it's a
why
whole
some.
lot more.
Yeah, oh, I know it's like exploded now. Which is I mean, I used to love video games, but I'm getting a little bit too. I got too much stuff to do for that now. Like I can't spend a lot of time doing video games. But I get like kind of bored a little bit too quick. But I know people love it. I used to.
How about a little bit of your long-term storage for your cultures?
Oh yeah, that was another one. I think I just made that video maybe like 2 weeks ago, but there are some well-established papers that were published back in the 1980s, and even one of the old Shroomery posts has some links that says some really obscure things that were published like back in the 1920s, I think. And then there's the thing with like somebody just posted, I saw the other day, where you kind of coax the mycelium onto filter paper that's been sterilized. I prefer, to be honest, there's slants, of course. That's kind of the traditional thing. You can just wrap up a petri dish with like cling wrap, the food wrap, like what, saran wrap, I guess most people call it
Yeah,
in North America. And I've fallen out of favor with parafilm. The problem with parafilm, it rips so easy. If you're gonna keep a culture for more than a few months, and then you go to use that culture, it'll almost definitely, the parafilm would have dried out, or whatever parafilm does, and it'll crack, and then your culture inside that plate will desiccate. It'll dry out, and it'll be, you'll have to revive it, which sometimes works, sometimes doesn't. The problem with parafilm, it's not the culture being desiccated or dried out. It's that when the parafilm cracks, it'll let contaminants in. So if you do have to reanimate that culture, it'll be contaminated. So that's the problem, you know. If you've got a petri dish and it's, or even a K cup or whatever, ketchup cup or whatever, if it gets air in it, you know, because there's not a proper seal around the outside, then it's going to get contaminated. So if you try to revive even a dehydrated culture, it'll be contaminated. So it's pretty much garbage anyway. But if you use the cling wrap, and now a lot of people are, I got on this grafting tape, but I notice you have to find the right brand of grafting tape. They have sort of different strength. I guess maybe if you were grafting, I don't know, like an eggplant or an apple tree, you might need a different strength of grafting tape. So the grafting tape, I've been through, I think, four different brands now or types, and you got to find the right one. And I hit on one that costs about is about $1 a roll, but the roll is like huge. It's long. I mean, you could wrap hundreds of plates with one of these rolls. It costs like $1.
And
And so that,
where do you find that at?
oh, just on Amazon or wherever your local, any
What's it
place that's
What's it called?
it's called grafting tape, like G R, yeah,
I'm gonna,
GRAFT. Graft is like a word they use when you make like a new kind of apple tree. Often they do. It's like if you cut off your arm and they sewed on another arm, like that arm would be like grafted onto you. So with fruit trees and things like that, they frequently do it when they're dealing with new cultivars. They want to essentially they do it with grapes and apples and citrus and everything. It's a very common. So when
I'm
you
gonna,
make that basically that little limb or that little bud, and you call what they call it grafting onto the new tree, you have to wrap it with some kind of protective tape, and it's essentially like fancy cling wrap. It's like cling wrap that's already cut for you, and that comes on a nice roll, and you just like use it. You don't have to cut it. I've got another video for cutting saran wrap that is like insanely dangerous because you have to basically take a huge knife, and like it's scary. So I saw some people use a hacksaw to do it.
I
Problem with
used a
that is you
skilled
get,
saw. I used an actual
yeah,
like electric saw to cut
oh,
my yeah.
you could use that. The problem is I had with that was the edge is kind of serrated, right?
Was it a sharp blade?
And
Like you used a sharp saw blade?
now that you're probably right. You need to use like one of those like tile blades or something like that, right?
Well, the
One of those
problem I
big.
found is that it kind of heats it up a little bit, so it starts to like as you're unrolling it, it gets stuck sometimes. So as you're going around your plates, it'll rip because it's getting. I don't know if it's melted in some spots.
Yep,
That's the problem I've been running into. So when you talk, go ahead.
yeah, that's the problem exactly. There's gonna be even that tiny little nick in the edge. It'll rip it, right?
Yeah. So when you're mentioning that grafting tape, I'm gonna check into that now.
Oh, for sure, man. If you find the right brand, it's like a lifesaver because I know that when you're you know, I do literally some days I'll go through hundreds of plates. Like and on a big day, I'll be going through at least 40 or 50 plates. And when you're getting down there to like that 40th plate and you're wrapping it and it keeps ripping, and I usually wrap my plates like 3 or 4 times, and because cling film's cheap, and so I might keep a plate for 6 months or whatever, and I don't want it to unravel and get contaminated. Um, so I essentially use agar plates for long-term storage. If you properly wrap an agar, so you guys got to forgive me for my pronunciation of agar. To be honest, in Michigan, everybody says auger. Cause you guys up there north of the border, if you're in Canada, y'all know about ice fishing, right? And an
Yeah.
auger is what you use to drill a hole in the ice. And so unfortunately that name stuck in my head. So for the past 25 years I've been calling it auger. And people now when I'm getting on these chats and stuff with people, they're like, "What the hell is auger? You mean agar? Agar?" And I'm like, "No, auger." And they're like, "How? What?" I'm like, yeah, auger. You what are you stupid? You auger. And they're like, no, and they don't know about ice fishing. So, I'm from Michigan. I know all about ice fishing. I used to
Yeah.
spend, every other day ice fishing in the winter. And yeah, so I've tried to slowly pronounce it agar or however the hell people say it.
Agar.
Agar or something. It's like, I don't know. I just sound like I have a speech impediment now when I say like agar. Like I'm throwing up or something. I don't know. So yeah, forgive me for my pronunciation. But
That's
just a side note, guys. Anybody who tells you there's a right way to pronounce something like a genus name like psilocybe or psilocybe or psilocybe, you can tell them to fuck straight off because there's not anybody who comes at you with that. Believe me, I've heard Russians, Chinese, Japanese say things like lactarius, lactarius milk mushrooms, and a Russian says it way different. Like they'll be like lactarius, and you'll be like, or like an Italian, lactarius, and you're like, what? You mean lactarius? And you're like, yeah, yeah, that, and you're saying the same exact Latin name. So anybody, like I said, anybody who goes at you and is like, "That's not the right way to say it," then you just tell them, "That's nice." Then bye. Like the thing is, like you'll learn. I've lived in Thailand for 18 years. When I speak Thai, I speak like a white dude. Like, and people look at me and they're like, "Oh, that's a white guy speaking Thai," but they still understand me. And so if they don't, then that's their problem, right?
That it
So
is.
I say it again. But they don't, you got to get over that. There's bigger things to worry about in life. If I'm ordering noodles and the noodle lady understands me, that's all she needs to do, right? Like she's not gonna be like, "Oh, I'm sorry, sir. You said kanom jin. It's supposed to be they're not gonna correct you. They're like, yeah, I understood you." So anyway, that's the idea. Agar. What the hell were we talking about? Long term storage or something
That's
like
it.
that.
Yeah, long
Yeah.
term.
Okay.
That was at the
Long.
beginning, though. I think that was at the beginning of the
Yeah.
conversation.
Tangent. I can go up on thousands. Water storage. Like, so if people find one of those videos, I've been using water vials and also what they call centrifuge tubes. And some people centrifuge tubes are normally 50 mLs, which is, I don't know, they're about 3 inches high, 3.5 inches high, and they're the size of like a big fat sausage, and they fit well in your hand and they're easy to work with, and they're getting cheap now, like a brand new PP5, polypropylene 5, the kind you can autoclave or PC, like you can put in your Instapot or your pressure cooker or whatever.
Yep.
You can literally like sterilize your media inside, like I guess do like what people call no pores. Is that what they call them now?
Yep. Yep.
So you make your media and you cap it up loosely and you just pressure cook it, whether it's sterile water or it's agar media, MEA, PDA, yeast, whatever the hell recipe you use. You just do it straight in the vial and you let it cool off. And if you're making slants, when you take them out, you just kind of tilt them a little bit and let them come back, room temperature, and try not to do it too quickly so you don't get too much condensation. And yeah, those slants, that's your traditional slant, and those work really, really, really well. And they only cost, I reckon, about 20 cents, like a quarter American. Like they're cheap. They're like cheap. If you're talking about a culture that you want to keep for, the next 5 years or 10 years, like they maybe not that long, but if you want to keep something or put it on the back burner for a year or two, you make a slant, you throw it in your fridge after it's grown out a little bit, and it'll probably be good for a year or two. The other thing that I like is that room temperature water storage. This is something like that a lot of the mycological and bacteria community has been doing for literally like decades. But for some reason, the hobby community hasn't caught on to it. And basically, what you do is you take any container. It could be an old whiskey bottle, but it should be the appropriate size. Sometimes little perfume bottles, things that are made of glass that might have a metal cap, that size, like when you buy perfume and it's got like, you don't put the little sprayer thing in it and it's got like a metal cap.
Yep.
Or I use these little drink bottles, like they're like 100 mil. like 4-ounce drink bottles. They might have like Red Bull kind of stuff in them, like those kind of sport drink things. Anything that you can pressure cook, PP5, glass, metal, and anything that's the appropriate size, you just put water in it, fill it up halfway with water, sterilize the water, and then take your plate and cut strips, like maybe quarter-inch, half-inch strips, and just put them in there, and literally cap it up, wrap the top with food film, cling wrap, saran wrap, grafting tape, whatever. Make sure you wrap it the same way as the threads, like tighty, righty tighty. Like wrap it the right way. Don't wrap it the opposite way. You want the threads and the lid to go down tighter, not like unloosen while you're wrapping the parafilm or the cling wrap or whatever you're using. That works brilliantly. We used to pull out cultures, I shit you not, that were like 10 years old, and they will start up on a plate in like 3 days. And here's the great thing, you can keep them at room temperature. So what happens, the mycelium sort of goes into, like, you might could call it hibernation. And as long as the strips of agar, they'll be just enough nutrition left in there to kind of, they go into almost this like deep sleep or like a hibernation. And as long as you don't mess with them, put them somewhere dark and somewhere where the water's not going to warm up or whatever. And man, like I just pulled one out the other day, a Moby Dick that was fucking 2 years old, over 2 years old, and it didn't it looked ugly, and I put it on the plate, and like 3 days later, bam, rhizomorphic mycelium. I was like, wow, okay, yeah, there you go. And they're essentially like bottles like that. They're like waste. They're like stuff you would recycle or throw away, right? So might as well use them for something.
That's interesting. Using the
Brilliant tech.
water. You think that's beneficial? Doing the
Oh
water
yeah,
stream. That's the
by
best way to
by
go.
by far, it is easily the easiest, most simple, most economical way. Even if you've got a culture, or maybe you wanted to do a clone or something, and you don't got time to get to it, just get it on a plate, make sure it's clean, and put it in a water vial and label it. You guys, make sure you label stuff. Come on, you guys. I know some people, might have short-term memory problems. I occasionally do too, but label stuff, you guys, please, for the love of all humanity, please, and your own sanity, like, please label stuff.
I'm not gonna lie. I got a few swabs sitting around that I don't know. I didn't label them. Now I'm like, fuck. What are they?
I know,
But
man. Well, that could be the next ape, or the next PE, like that could be the next toke, or fucking whatever yellow, whatever the hell they're sending out now. Like, come on, man. You got to just a Sharpie costs like $1.
so should I just throw a name on it and send it out? Is that what you would do? Just throw any name
Yeah,
on
you
it?
could start. Yeah, just get name it after some alien or some shit in some sci-fi movie and be like, yeah, this is the Klingon 4682 or whatever.
And
Seen
then just
Klingon
throw it out there, eh?
like, yeah, be like, yeah. Just make sure you sell it for like $50. People will think it's like the bomb.
That's
Like,
cheap
wow,
nowadays.
is
That's
it
cheap.
really? People have been telling me this craziness about like swabs going for $50. I'm like, really?
uh,
Whew. I
what
sell
is it?
I guess I'll put in a shameless plug here. I do have, on the Spore Swaps, I do have a lot of my stuff, the sporeswaps.com or whatever it is. But
Yeah,
I sell them for $15, but the only reason I want to get my genetics out there, because I've put a lot of effort into making these crosses, and a lot of like kind of money too, to be honest. So just to like sort of recoup some of my losses, well, then I wouldn't really call them losses, but my costs, $15 for a pair of swabs. And the only thing is, that's just basically get my ass to the post office and put them in put them in an envelope, because I gotta buy the damn greeting card and all this stuff. So it's like pretty minimal. I'd love to give them away. I mean, if I was over there, I would just be handing them out, like, at the mall or whatever. I'd be like, hey man, do you want a F2 cross of like you know PE cross Moby Dick? Do you want one? I'd just be handing them out at like the kiosk at the mall, or whatever. But no. But yeah, I gotta try to, like, at least, my time and effort. But yeah, there's tons of stuff on there. I managed to get some monos, some spores from one of the iceberg things that one of my friends sent me. It's one of these other clonal cultures. There's all kinds of stuff. I heard people selling monos now for like 150, $200 too.
No,
And it's craziness, dude. Like, I don't even
I've
know
never
what's
heard
going
that.
I
People are buying it though. Like, are they buying
Well,
it?
that's I don't know. That's my one of my goals in life is to teach before I die. I want everybody who's listening to isolate their own monos. You can isolate monos from any spore print, any swab. If people listening get into my videos, like really watch them. I've got one of them I call a ghetto dilution. It's essentially a cereal dilution that you use to like glass sport drink bottles, and you essentially can make. There's another one people are calling a grab and drag, where I take basically a swab, and with a flame sterilized what do they call it, forceps, the tweezers, you just grab a part of the swab, like a tiny little bit of the black bit, or the colored part, or if it's an albino, like I did it with an albino gumby a few weeks ago, worked like a charm. You essentially you draw like sort of an S shape, like a serpentine, like back and forth. But what you do is you grab a bit of the swab and you jam it into the agar surface, and then you kind of like keep it in the surface, and as you're kind of keeping it pegged down under the agar surface, just like go like right, and then do a little S, and then like hockey, yeah, when you're like that, you're like shoot back and forth, and you make a little S, like a little serpentine, maybe like two or three S's on there, and you can pull monos from one plate. Like, it's easy to get monos, man. You guys, it's really, really easy to get monos. The problem is the verification of them. You do, I hate to say it, but you do need a microscope, to 100% make sure there's no clamps there, which is what a mono is going to be. Um, you're going to need a microscope. But, if you're at a school or a university, or have access to, your kid's freaking science classroom at the school, they've got a microscope laying around. You only need 400x. You
There,
go in there for 5 minutes. Ask your kid's teacher, like, hey, do you guys got like a microscope laying around? They'll be
you were
like,
cutting out
yeah.
there for a minute again.
Sorry, man, I can't do anything about that. That's the
No, no,
internet.
that's good. I was just letting you know so you know.
Oh yeah, so I
You
can
know,
fill in the blanks. Like,
Blue Michael, right? He made a comment saying people are doing mono work for 1 to 2K. That people are charging that, I guess, to get monos.
yeah, yeah, I have seen that post, and I almost, like, my jaw dropped. I was like, what is this? What are these people thinking?
So really, they're just charging for a service to get a mono. Is it like
Yeah,
I've never heard of this? This is all new to me. I've never heard this. So
to be completely honest, that was what kind of provoked me to make a lot of these videos, because I made monos. I don't What I did for my PhD was essentially based on the biological species concept, which essentially says that if you can mate two haploid spores together, then it's the same species. It's kind of like when you take two dogs and you put them together and they make another dog, right? You call them all dogs, right? So if you take one cube and you mix it with another cube, or something that people are calling natalensis, and you mix that with another cube and it mates with that other cube, then it's not another species. Simple as that.
it's still a
Like,
cube.
that's is still a cube, and that statement right there is probably the most controversial thing I'll say during this whole podcast. But if you can mate things together, like if you took a Chihuahua and a Great Dane and you put them together in the right conditions, they could mate with each other, right? They look different. They look very, very different, but they can make another dog. It wouldn't be a Great Dane and it wouldn't be a Chihuahua, but it would be another dog, and everybody would recognize it as another dog. You would feed it dog food, right? You'd keep it in a dog cage. It's a dog, right? So for some reason, these people have gotten it in their head that if I take, oh, I don't know, something I'm going to call a cube, and I mix it with something I'm going to call natalensis, and I mix it together, that's no longer an adellensis or a cube, it's a hybrid, and this is not correct. And that statement has gotten me on the bad side of a lot of people, and I don't care because it's it's what I've done for like the last 20 years of my life, so I know a little bit about it.
Oh, hey, thanks for speaking the truth. You know, that's what we're here for. And Blue Michael also says thanks. He appreciates you for saying that. I know you can't see the comments and stuff. That's why I'm repeating them back to you.
Oh yeah, same here. I can't. I'm like I don't want to mess with my computer or the phone. I'm just gonna
No, no,
let it
that's
roll
good.
like ride like it is because it's bad enough the internet's cutting out. If I start messing with the things, I don't want to.
P Funk's telling you to preach. Keep like preach. You preach, Ed. Preach.
Yeah, some of those guys I'm familiar with some of them already, and yeah, they know they're familiar with my stance on this, and I'm trying to be. You're not maybe quite as familiar, but I'm being nice right now. Believe it or not, like I'm being quite nice about it. But my patience with certain people or groups is wearing very thin because I feel that like people are getting robbed, they're getting scammed, and I'm going to say something controversial that'll probably piss some people off, but my job right now for the rest of my life is not to make people happy or to praise other people's or assuage their egos, to be like, you're the greatest in the world, you made a fucking mono. Like my job is to tell every one of your listeners that you can do exactly what they did. Here's what you do: go to YouTube and watch 3 of Ed's videos. They'll take about 30 minutes. Get yourself a fucking $200 microscope or borrow one from your friend or use your kids at the school. And you too can produce monos, and then you can do crosses, and you don't need to worry about verification processes, which also cost hundreds, if not thousands of dollars. Like, let's verify my mono and let's verify my cross. Well, if you have two well established cultigens or strains and you isolate monos from them, you don't need to verify them, right? Because if you have, I mean, assuming it's labeled properly and it's not doing some weird revert thing or something, like, if I get a mono from a PE, I know that's a mono because I got it, and like I can use it for crosses, and then that completely eliminates the need to pay for some kind of verification process,
So the
which is
best thing to
something.
do, because I'm just getting into this actually. I bought the scope. I'm doing the serial dilution, like following your videos, and so when I get my monos, I just keep them on a plate, labeled M1 whatever, and just
Yeah,
store those, and that's a mono forever, is what you're
yeah,
saying.
yeah. Yep, exactly. I accidentally, again, how I sort of serendipity, I believe, is what they use for it. I was fruiting an MVP. I thought I was fruiting an MVP, and somebody mentioned this earlier. I forgot who. You cannot fruit monos, and I realized this after I on my third MVP shoebox, and I think I even had a bag, and I'm like, why will this not fruit? And then finally, I pulled a little tuft off one of the shoe boxes, and I looked at it in the scope and like, oh my god, this is a mono. Like I tried to fruit a mono carry on for like 3 months, and I'm like, why is this bitch not fruiting? And I was getting really, really upset. And I was like, the plate looks kind of, they have this like pseudo rhizomorphic growth. And I'm like, the plate looks pretty good. And then I looked at the plate also. I'm like, oh my God, there's not a clamp in sight. This is a mono.
You know what?
So
Now that sorry to cut you off. You're just getting me excited here. Now that you say
no,
that, I've been trying to fruit these penis and albino penis envies and these
yeah,
starry nights for like 4 months now, 5 months, and they just will not grow. Like they will not grow. It doesn't matter what I do, how I do it, it will not grow. So
dude, you
should I
need to
go?
get out. You should, man. It happened to be not once but 3 times. I was like, even after that first MVP mono that I discovered after I tried to fruit it 3 times, I was like, oh my gosh. And then I had I think it was a Moby Dick culture, and I'm like, oh my god, this is a fucking mono too. So I ended up in an afternoon with a microscope. I was like, oh wait, now I got like three monos. I guess I should mate them together. I was like, oh my god. So, you know what I do now when I do a multi-spore? I make damn sure it's a dikaryon. If I get a swab and I'm trying to just get a dikaryon for fruiting, because dikaryons are the only one. Like monos will not fruit. So here's another little trick, just kind of a side note. If you think you've got a mono and it sits on a plate for months and months and months and you don't see a fruit body, like even a little pin, it's probably a mono. But if you don't want to wait that long, you need the scope because you got to look for the absence of the clamps. But if you basically have a plate like my MVP plate, and I look back through a stack of like six plates or seven, and I was like, wow, there's no pins. Because a dikaryon cube culture, almost all of them, they will pin after about 2 or 3 months. They'll pin on agar. So if you don't see a pin after like 3 months on your agar plate, you can assume that it could be sterile, which occasionally happens, but we don't really need to worry about that, to be honest. That's a whole mating allele discussion that we don't need to have right now. It's a more complicated story. But if you don't see, like, if you think you've got a dikaryon and a plate sitting there for 3 months and you don't see any pins on it, something's wrong. Something's probably wrong. When I cross monos, also kind of an extension of that. If you cross monos and you do the cross, they will form a fruit body at that interface or one side of the interface. It's again another kind of complicated topic, but if you don't see a fruit body, it means maybe the cross wasn't successful, or maybe even one of what you thought were monos might have been a contaminant, or the whole culture got contaminated. But if you leave any dikaryon cube culture on a plate for like 3 months, you will see pins almost definitely. You will see a little pin on there. And then if you do see a pin sub that, like you can clone that. You can just clone that pin straight from the onto a new plate. And then you'll know you've got a fruiting culture.
Huh.
But going back to the monos, yeah, monos are exceedingly easy to get. You need to make sure that if you're not going to waste time. Okay, so if you're isolating a mono from my little grab and drag technique, or some people are calling it the grand drag. I don't know if or the grand. I don't know. Some people have been coming up with funny names for it. I don't know where. I don't know. I just did it. I was messing around one day. I didn't think about it that much, but you know that there's a quote that goes back to Louis Pasteur. I didn't realize it, but I looked it up the other day. That chance favors the prepared mind. The more you read, I read a lot about cannabis tissue culture just because I'm interested in it, and legalized here, so I'm really, really fascinated in what people are doing. I read a lot about viruses. I read a lot about like normal edible plants. I read about like all this stuff, and you'll be surprised. You'll see things in other literature and other books, or even on social media, where you're like, oh, like wow, like I didn't think about that. And it just like it's good just to like kind of read random stuff that I mean, I hate to say, you can go on Facebook groups, man. There's a lot of people posting seems like random trivial stuff, but you'll get like little gems out of there, like, oh, like there's some people out there that I'm sure are they know way more than me. I'm, to be honest, getting into this breeding thing or whatever you want to call it. Like, I don't even really consider myself a mycologist per se. I'm just somebody. I'm like a glorified farmer, you know. But if people want to call themselves mycologists, that's cool. But me, I don't really. I don't know. I'm just like a fancy farmer.
Blue Michael had a question I was going to get to you. He says, can't monos revert to dicaryons?
No, never. So I saw somebody post that the other day, and no, a mono will. So once a culture is monokaryotic, the only way it's ever going to become dikaryotic is if it encounters another spore, another piece of mycelium, or another dikaryon. So there's something called the Buller phenomenon, which is well established, but it's not the best way to go about breeding. So, whoa, this is getting deep, dude. So, like when people have dicaryons, they've been doing the DDK, the de-dicaryotization. They call it DDK, and that's a whole other procedure. That's Comes from a book. There's a book from by Chang and Miles, or Chang and Miles is the way they say Chang here, but it's the name of an elephant. The Thai word is Chang for elephant. But Chang and Miles, there's a book they wrote. I can't remember the title, but it was published in the '80s that illustrates the DDK, the de-dicaryization, quite well. So if people want to look that up, it's not a new technique. But anyway, so you need monos. If you put two monos, so monos are what are called haploids. Haploids are the equivalent of like human sperm. So for, let's just say about humans, to make a new human, you need a sperm and an egg. So the egg is haploid and the sperm is haploid. Now, if you put two sperm together on a plate, they're not going to do anything, right? But our fungi are a little bit different and they don't really have a sex, you know? So a sperm and a sperm can make essentially what would be what we call a dikaryon. So it would be it'd be a metaphor analogous to like a sperm and an egg. So like if you're a woman and you only have eggs, you can't make more humans. And if you're a man and you've only got sperm, which are haploid also, you can't make more babies. You need two haploids to make the dikaryon, or in the case of humans, diploid. Like some of the bio people out there, they're like N and 2N. They usually. So the short answer is no. A mono will never become a dikaryon by itself. That's the short answer. Um, Dealing with mono, if you're dealing with a mono isolated on a plate, it will never, ever, ever become a dikaryon, unless you have maybe accidentally pulled off another spore, or you've got maybe a mixed culture. If it's coming from a multi-spore plate, that's why when you do the grab and drag technique, like I was saying about the swab, like grabbing a bit of you have to really, really verify that you have a mono. If you start trying to mate dicaryons with monos, you're getting into the realm of what's called the Buller phenomenon, and that is like another 2 hours of explanation that I don't want to get into. Buller, Buller. If you look up on the internet, just look up Buller phenomenon, and you'll see lots of stuff about it. It's well studied. Another thing that they've known for 50 years. There's a guy named Raper who goes into excruciating detail in one of his books that was written in the late '70s, I think, about it. And it's named after a guy Buller who was a mycologist. I believe he was a French mycologist back in like the 1890s or something like that. So it's not a new thing. Like all these things like DDK, Buller phenomenon, these are not new things, you guys. You may have never heard of them, but they exist. They have existed for a very, very long time in the literature. Um, Supplements. I know people wanted it. Somebody wanted to talk about supplements too. That's all out there, you guys. The Chinese have figured out how to grow mushrooms really, really, really well. Like, so if you can read Chinese, you can figure out how to do that. But if you can't read Chinese, you just wait for some dude or Google Translate, like erythritol and azomite and all these things like D3. These are all well known, like Chinese secrets. But like we're just learning about them, in the cube community. But again, if you're just growing cubes, look in a book about like pleurotus, oyster mushrooms, or agaricus, the white button mushrooms. Like you'd be amazed at the kind of little secrets you can find, that'll apply to cubes, that they might not be on Reddit. you know, like all the secrets about growing mushrooms aren't on Reddit yet. Surprise,
I don't think
surprise.
there's many secrets there. I venture there quite frequently, and it's the same thing over and over and over again.
Yeah, that's the one. I gave up on that one already. I was on it for like a week, and I'm like, no, 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 this is gonna kill my brain.
It's not the right place to be. I want to touch on that monokaryon for a second there. I was told like once you do a streak plate, right, with your swab, if you get a culture that's growing away from the streak, like shows up away from it, then it might be a mono. Do you know what I'm saying here?
Yeah, I do. It's actually the opposite. If you get if you get okay, let's see. So let's say a streak plate or a multi spore. if you see generally something that kind of wants to like climb over part of the rest of the culture, it'll be rhizomorphic. So rhizomorphic literally means like a root. That's it's like the Latin name rhizo means root, like a mycorrhizal fungi, things like that. It means so if you get something that looks almost like a piece of cotton string, that's a rhizomorph. And when you see those pretty pictures of agar plates that have that nice concentric sort of like starburst pattern, those are the rhizomorphic plates, and that's a good indication. Not 100%. It even tricked me with that MVP. It's a good indication that you probably got a dikaryon. So if you're trying to pull a dikaryon, what you want to do is take a little tweezers, like a little cosmetics, like eyebrow tweezers, and pull that rhizomorph off and put it onto a new plate. And you got to be patient because you're literally pulling off like maybe a 16th of an inch piece of that rhizomorph. Make sure you let the tweezers cool off and pull that off and just mash it down into a new plate. And within a week or two, you should have a nice, really pretty rhizomorphic. You can post it on Art of Agar Osgard or whatever that Facebook thing is, and put it on your Instagram, and people will be like, "Wow, look how pretty that is." Now the opposite, if you're trying to pull monos tend to be more irregular in their growth patterns, and it's not, unfortunately, the morphology, as they call it, the overall appearance of what it looks like on a plate is very, very different, but it's not as recognizable. It goes, I've got some monos that I've literally subcultured or transferred like 15 times. You can keep them going. Once you get a mono, you just keep subculturing it just like you would a dikaryon. And their morphology varies a lot, way more. Like the same the same monos that I've used for dozens of crosses. When I subculture them, when I do a cross, I set up the crosses and I'll subculture to a new clean plate so that I have the mono for perpetuity, forever.
Yeah.
Uh, yeah, basically they look really different every time they get on a new plate, and I don't it's a little like it's almost a little bit like it makes you really nervous. But as far as pulling monos from a multi-spore plate, you kind of gotta use your luck and into I hate to say it, but intuition. And the one thing is, when you're making transfers, make sure you try to use as small of a transfer piece as possible. So whether you're trying to get a monoculture, like i.e. like a dikaryon that's isolated in a single genetic, you want to pull off with the tweezers or cut with a scalpel, like the smallest tiny little piece you can. I mean, something that's literally like, the size of a grain of rice, like uncooked rice, like that size. That's what you want to be transferring. I see a lot of people, they'll transfer these big chunks, like half inch, 3/4 inch chunks of agar to a new plate. That's not, you shouldn't do that. You wanna if you especially if you're on a T1, T2, T3, when you go to a new plate, a transfer plate, that's what T stands for, right? When you're doing that, you want to take as small a piece as possible. Not minuscule. I mean, I'm not talking like ballpoint pen ties, but a rice grain, or maybe a little bit bigger than a rice grain. Let's say like I don't know, like a grain of wheat or millet. Some of you guys know millet or Milo, right? Like that's the kind of size piece you want to be transferring to the next plate. So that's where like the scalpel blades. I love those like 11 scalpel blades with the number 7 handles. Those are brilliant. But if you're using maybe, like a paring knife or something, it's probably not the best tool for doing transfers. Something you'd like, core an apple with or peel an apple with, probably not the best thing to be doing.
That's what I
Is there any?
use.
Yeah, you know, I used to drive a 1985 Chevette that had like, 2 cylinders out, but I got
Well,
now I drive a 125cc motorcycle, so it's not really like an upgrade, but it's
I guess
newer.
I gotta
But no,
I gotta put
if you're gonna,
my pitter away. It's gonna go away now, man. That was my tool.
I know, well, Christmas is only like 10 months away, You drop like subtle hints like, hey, you leave that Amazon page open on the computer for an extended period of time and be like, look. And then yeah, then your partner or whatever will be like, oh, you're a psycho. Are you gonna kill me? Like, why are you looking at are you gonna like dissect me after you kill me?
That's it, eh? We've been watching a lot of Grey's Anatomy, so maybe a surgery's in order, you know?
It could do, but even if you're gonna do that, you should have the right tools. So it could be an investment, multitasker.
That's it. Um,
Just burn
Trying to
off
think.
the blood when you're done. You got your alcohol lamp. You can get rid of the DNA in the blood. The blood for like you gotta just burn it. 70% ethanol and get rid of all the DNA on the blood and hair follicles and whatnot. So sorry, yeah, I don't mean to I would love to go back to that mono question, but when there's questions like that, it gets quite specific really, really quick. So it's hard to answer a question like how do I get a mono because there's a lot of different ways, and also it's not always the same. Like every little situation, like a mono will look different on different media, and it'll also look different. Like temperature and light can affect them, and so you'll end up getting monos that they'll look like this toast kind of fuzzy like patch on a plate, and you'll be like that does not look right, that just does not look right. It doesn't look clean. Maybe it's contaminated. And then I went through this for like 2 or 3 months, and when I finally did my to be honest, you got to be patient. If you get monos and you mate them together, even as you're mating them, you're like, these might not be monos. And then later on, you'll take that little section where they meet and you subculture that, and that's where you'll see that dikaryon pop out, and you'll be like that rhizomorphic dikaryon, and you'll be like, oh my gosh, I think that worked. Like when you put two monos on a plate, say a half inch apart, you take two little chunks of agar from each mono, and you put them on a plate and you let them grow together for a week or two. And then at that interface, like weird things will start to happen. And occasionally near that interface, or maybe on either side of that, you will see a fruit body. But that usually takes 2 months, 3 months, and most people aren't that patient. So what you'll do is where they interface, you subculture that, and then on that new subcultured transfer plate, you will later, after it grows out, you check for clamps. Then if you see clamps, you know that your two confirmed monos that were on the first plate, they were monos because you look back and you look at the original cultures and you're like, no clamps on that mono, no clamps on that mono, and now I have a dikaryon that has clamps. And so that's how you verify your cross. And then you make your grain spawn, and then maybe 2 3 weeks later you put that to sub, and then 2 weeks later you got fungi. Those fungi produce what are called the F2. So it's a little the terminology is a little bit weird with fungi because so when you do an initial cross between two monos, that's what in botany is called an F1. So F1, F stands for filial. It means the next, like the brothers is what it means in Latin. So if you see an F1, F1 means it means that it's not like the Formula One. It means the first filial generation, and that's when you take two monos, the parents, and you cross them together, their offspring. So you are the F1 generation of your mom and dad, right? So that's the way the terminology works. So if you had a child with another woman, I'm assuming, or a childbearing person or whatever they're called now, if you had that would be it's like your kids are like the F2 from your mom and dad. So the kids' grandparents, like the grandchildren, the grandchildren are the F2. They're the second filial generation from your parents, which would be the child's grandparents, right? Does that make sense?
Yeah, yeah. So it's from like going from spore to clone, spore to clone, or
No, no, no.
clone to spore.
This is where people get a little confused. So yeah, there's no cloning involved. The only reason you clone is to preserve. So if you do get an F1 that looks nice, you might want to clone it. Cloning is just basically to keep that genetic lineage in circulation. So if you do happen to you take two monos, or even if So cloning is just what if people want to get like canopies, you just clone your nice cluster and you take one of those fruits and you clone a good one. So cloning is what they do with like bananas and apple trees and roses. And when you go and you see like the same kind of rose at Walmart or whatever, but you see 1,000 of them,
Yeah.
those are clones. Those are all clones. So they do it in tissue culture, very similar to what we do with our mushrooms. But those are clones. But the idea with breeding is you don't really want clones because you're looking for something different, right?
So you're just going spores, spores, spores. Keep going to the spores,
Yeah.
and
Yes, exactly. So in order to get new spores, you have to make a fruit. So the sole purpose of you taking two monos and putting them together on a plate is to either make that F1 or to take that F1 and get the spores from that, and then that becomes your F2. And then you take that F2, you go back to spores, and then you do the whole process again and again and again. This is why when people talk about, oh, this is an F7 or an F8, that could take years to do it. In plants, it could take literally like decades to do that. So people who are breeding tulips, 200-300 years ago in Holland, there's a famous story about the tulip mania, and that's a whole other story. But yeah, it's a lengthy process. Now, if you get lucky and you do like a ghetto swab, double swab, you
yeah.
do that, you do a multi-spore, then you're talking about basically a different kind of mentality. Like you might get lucky and you pull a pheno from that. Well, so this is where the terminology gets a bit deep. If you're doing multi-spore and it's from a ghetto swab, you could just simply get one of the parents. Because if you take two spores and you made them together, once that dikaryon forms, whether it's on a multi-spore plate or whether it's on a plate that you've set up where you've actually put two monos on, once that dikaryon forms, that's pretty much the end result. That's like saying like you can't turn into your mom's egg and your dad's sperm, right? It's not going to happen. Like you can't go backwards. This is like a one-way path, right? So if you want to get you again, like a clone of you or another one of you, you'd have to have exactly the same sperm and exactly the same egg. Now you probably got brothers and sisters, right? Your brother's not the same. The reason why is because your dad's sperm were all different, right? So you see where I'm going with this? If you've got a multi-spore plate and there's 1,000 different spores on there, that means you have 1,000, and let's assuming all of those 1,000 have the potential to mate with that one other spore, which in this like metaphor would be your mom's egg. Like there's 1,000 different brothers and sisters you could have, right? And they're not all the same. So this is where the numbers and basically the manual labor gets a little bit intensive because if you're doing multi spores and that's your breeding strategy, i.e., you're doing a double swabbing thing, the outcome is far more unpredictable.
Then by
That's the
doing it
long.
with the monos.
So yeah, that's the short story is that monos allow you to be more precise.
Yeah.
And there's like you mentioned, once you get a mono, you can grow it forever. I think I said that wrong. In perpetuity. You can grow it for once you get a mono, you can subculture that thing, or I say subculture because I worked in like labs where I grew other stuff. They say subculture. You can keep growing it again and again, transfer it over and over and over, put it on a slant, put it in the fridge. So when you get a mono, even if you don't have time now or not next year, maybe 5 years from now, you pull it out, you regrow it, and you've got your mono still. And so what I do now, I got about 15 monos on stock. And last week I happened to get spores from iceberg. And last week I happened to get spores from a BHT. And like I made monos from them. So now all the 15 previous monos I have, I can take my new iceberg mono and I can mate it. So I now can do 15 crosses like
That's
straight
amazing.
away. Like literally in an afternoon, what I'm probably going to do tomorrow, I'm going to make 15 new F1 crosses. Now,
And that's what people are paying this crazy money for is those monos. Is that's going back to like the thousands of dollars or whatever they're paying for these monos. Like people are charging for those monos. Going back to that, which we already covered, but
Right. I can tell you why they're doing that, because they know that once that mono gets out into circulation, what are you going to do? Share it with your friend, right? That's
but I
why the
mean,
$50 that's why the $50 swabs are going for $50, because they know as soon as somebody, like in a month, I mean everybody knows this example, the toke, right? Like they've been sitting on that for like 2 years, okay? As soon as my friend got a toke swab, what did they do? Ripped off a piece of it, plated it, and sent the rest of the swab to me. And that's exactly what everybody else will do, right? You
but isn't
get
that the
two
point
swabs.
though? Like, isn't that the point? Like, I don't know. Maybe I'm looking into this wrong. That's the point though, to share it. No.
Yes, from my point of view, because I don't need to make money off of these things. But if you spent two, so this goes back to this is why we have patents. And this goes into a whole ethical, like, say you've got a disease, a drug that cures cancer. Now, the ethical thing would be to release that drug to cure everybody in the world of cancer, right? See,
Correct.
we're on the same page. But what if that company that spent $50 billion to develop that drug makes no money from it, right?
Yeah, it makes
Would they
sense.
develop would they develop that drug if they knew there was going to be no return on their $50 billion investment? I mean, it's a
No, no.
simple answer. No, no, of course not. I mean, that's a redundant. That's like a rhetorical question. No company that is going to be successful is going to spend $50 billion to develop a drug and then not have to be able to make money off it, right? So if I spend 5 years of my life, not me, I'm just saying as an example, somebody like me, if I spend 5 years of my life developing a new strain or a new cultigen and I release it, I've got basically one single chance to make all my money. And then after that 2 day period, when I've sent out all those spores, that window closed, I have just eliminated myself as part of that market, right?
Because you sent it out, and now people are just sending it out into the masses.
Yeah, and that's exactly what I would do, and you would do, and every other person out there would do, right? Like I wasn't on the waiting list or whatever, but I'm gonna have a toe swab. Somebody's sending me one. It's in my mailbox right now.
Never even heard of it. I've never heard of that.
Oh well, I'm sure some of the listeners have. It's a kind of it's been the cultigen that everybody's been sort of waiting on for like 2 years. So it's a tricky thing, and that's why we have like patents on medicines. You know, you can go to the store now and buy generic aspirin and ibuprofen and whatever for headaches and birth control or whatever. But some company paid a lot of money in research, R&D, and lawyers and whatnot to get that drug to market. So it's a big. I don't even think we need to go there, but it's the same idea. Like if I So this is why I got into this whole mating thing, was because I kind of wanted to eliminate this problem. Like almost like my weird backwards way of going about this problem that we have is that if you teach everybody to make monos, this problem goes away because you don't have to pay for $50 spore swabs anymore because you can make them at home by yourself. Right? Like this is it's one of those examples, I hate to bring up the like, you give a man a fish, you feed him for a day. You teach him how to fish, you feed him for the rest of his life.
Yep,
Right?
that's great.
I don't want to go all like biblical and shit, but everybody knows that, right? You teach somebody how to do something and you give them the skill and they don't need and it eliminate so in that whole process, not only have you've raised up people's knowledge level, but you've also given them a skill that they can apply to other situations. So if you've got the skill to sterilely isolate monokarons and mate them on an agar plate, you can pretty much do anything in mycology. So this could apply to shiitakes or oyster mushrooms or cordyceps or whatever. Like once you learn sterile tissue culture, if you're to the point where you're isolating monos, you are so far ahead of the curve from other hobbyists. And I know people out there, they're shooting LC into grain bags. You buy a grain bag off for $50. You buy another, LC syringe for $20. That's cool. I'm glad people can do that. But I assume that your listeners are probably, most of them are well past that level of knowledge. But that's a good place to start. I'm not saying anything bad about that. But at some point, people are gonna get those mushrooms and be like, "Wow, this thing's got spores," and then they're gonna be like, I can make like spore prints," and they'll be like, "Oh wait, I can clone this," and they'll be like, "Oh wait, I can make my own LC." And then they'll be like, after they get that LC, and they're like, "What the hell have I gotten myself into?" Like when you
You would
start
hope
making LC, you'll be like,
you would hope
"Why
they
the?"
get there. Right?
Oh, they will. Making LC is dead easy, man. Like I'm not trying to promote my YouTube channel. I get zero monetary value from that. I don't care about that. I'm trying to get people out there. I've always been anti-government. If you can't guess, if you can't guess, like I grew up listening to like Public Enemy and like NWA and like Slayer. Like I've always been like fuck the man, fuck the government, fuck authority. It's like some of like that Rage Against the Machine. It's like fuck you, I won't do what you tell me, that kind of stuff. It's like that kind of sentiment. Like I grew up listening to like Dead Kennedys and like Black Flag. Like you know I've always like not really like people telling me what to do. So I'm sure a lot of the listeners out there probably down with that sentiment. And so if I can teach somebody or at least help them along the way to learn something that essentially frees them from the tyranny of the government and the system and the gatekeepers, I hate that word, but whoever the gatekeepers are, if I can give them any little bit of knowledge to help them fight. We used to call it the man, the government, the federal, whoever it might be. The man is a different person for everybody, but fighting the man, that's like my goal in life. Basically, is like fuck the man. So that's it. If you're really
So
contemplating
what you're
spending
saying
your
technically is you want to fight the companies out there that are char-
yeah, any kind of authority, any authority. It could be your mom, could be your dad, could be Monsanto, could be some other Facebook group. It could be some. It could be what I don't know. It could be some of the. It could be I don't know. It could be your neighbor. Yeah, it just the general anti. What do they call it? Anti-authority. I don't know. I'm not too extreme. You got to live in the real world. You can't go around like fucking your own life up because you're trying to hate on other people.
No, you're not
There's
hating.
a certain no. There's just a certain bit of distrust you got to have for when people try to tell you what you should do. You have a little like healthy bit of distrust and kind of anti-authoritarianism
And I
there.
mean, like you said,
Like
once you learn that for yourself, you know what you're getting. You're not ordering a swab that's saying, blue meanie or whatever, and getting something totally different. Eventually, you know exactly what you're getting.
yeah, exactly. You just make your own. That's funny you mentioned Blue Meanie because that was what started me on this path a couple years ago. Was Blue Meanie is actually the colloquial name in outside of America for pans,
Yep.
pancyans, Paniolus
Yep.
cyanensis. And I don't know who came up with that name Blue Meanie for a cube, but I really, really, really wish they wouldn't have named it that, because it's a cube. Blue Meanies are pans, right? And I don't know, it was a marketing thing. Again, they knew that people would get confused. Either that or they were very, very ignorant about mushroom names. And I think they knew that people would think, oh, pans are stronger. Like pans are
Oh,
stronger than cubes. But if I name a cube after this stronger mushroom, like everybody wants apes. And I don't know, everybody at least kind of realizes that apes are no different than cube. Apes are just a white cube. Like there's no scientific basis, HPLC testing or whatever, which is also flawed and also biased and also like a whole other topic I don't want to get into. Let's just like let's just piss off like one group of people per podcast. Like next time we can go after the HPLC people. Um,
Why stop at one, man? Let's get it done.
no, well, I can come on again. Don't worry. I've got plenty of other people that
Controversy.
I'm gonna be like, I'm gonna get like hate mail and stuff. After this, they'll be like, "Hey, give me your address so I can send you some new spores," and they'll send like a letter bomb or something.
Anthrax.
Yeah, they'll go out like old school, like Unabomber shit on me. Like I'll be like, "Oh, look, it's my new cultures," and it'll be like a bomb. Like, oh wait, that would be me doing that. Oh wait, no.
Censorship. Censor.
Yeah, it's all good. But so I was wondering, is there like you got a time limit? What did we start at? Like 2 hour and a half?
Yeah, it's been an hour and 35 minutes.
Oh, okay. Yeah, I couldn't remember. I'm on a different time. Did you want to go on to another question? Like, I said, I can go off on tangents, and maybe I don't want to piss off too many people, like in this podcast.
Nah, I was
I'm trying to be nice,
I was
man. I'm trying to be
I was
nice.
just going with the
I don't want
conversation
to.
there. We weren't we were just going with the conversation. That's all.
Yeah, it's cool. That's the thing. You got to get a thick skin in this whatever you want to call it, hobby, man, this endeavor. You got to get a pretty thick skin because there're going to be haters and there's going to be scammers and there's going to be just people out there looking to make a quick buck. And there's going to be, I hate to say it, but misinformation. Some people purposely put out misinformation to kind of throw you off the trail. This whole mono story, like the simple fact that we're talking about it right now, it kind of blows me away because 20 years ago I was literally in a lab picking spores off an agar plate. We did it a different way. Like I don't. Maybe I'll make another video about it. But the easiest way are the three ways that I have on my YouTube are the easiest ways by far for a home cultivator to do it. But there's other ways. But yeah, I was doing this like 20 years ago, and when I figured out that the MVP was a mono. And I'm like, oh my gosh, like I accidentally did this. And then I kind of didn't realize because I used to use them for what are called mating studies. Basically, when you find a new species, there's two types of mating systems in basidiomycete mushrooms. They're either called like bipolar or tetrapolar. And in order to figure that out, which is a whole other topic, you have to mate single spores with each other. And this is where the discussion of, you probably heard mating alleles and A1, A2, B1, B2, pheromones, stuff like this. That's like not necessary. But if you get into that in more detail, you gotta go a little bit deeper into it, and but we don't need to go there. But I didn't realize, but I was doing this 20 years ago. Like this was common procedure, and when I realized that this was such a highly like secretive thing that people did, I was like, why is it such a secret? Like, why are these people gatekeeping essentially? If that's, I'm not sure if I still don't know what gaslighting means, but gatekeeping, and I don't know what based means either, but I kind of know. But I was like, why is this not common knowledge? I was just like wondering. I'm like, why is there not like a YouTube video about this? And I'm like, okay, well, I guess I need to make one. And when people first saw it, they were like, "Oh my god, like I never knew this." And I'm like, "I thought this was like normal stuff that people did." Like, I don't know. But the gatekeeping thing, that whole shit about snake venom is like I want to punch somebody in the face every time I hear that.
I actually I heard about that. I can't remember where, but I heard about that on
Anytime you hear
some interview or something. I heard about that, and
yeah, I saw it advertised yesterday as a, yeah, I don't even want to get into it. There is no need to use snake venom. Snake venom, if you are going to do so the new slant, it used to be that if you were going to breed cubes together, you needed to somehow make the cell walls permeable for the anastomosis and the somatogamy and blah, 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 blah. All these fancy words that people, like, oh, well, that sounds fancy. Like, they use all these words. Essentially, if you got two cube spores together, they will fuck, right? You put them on a plate and they will fuck each other. That simple as. and they were trying to make it all like you got to have berry white and like candles in the background, like you need snake venom to get like spores to mingle and mate with each other. It's like, no, you don't. You don't need a bottle of Chianti, berry white, and candles. You just put them on a plate next to each other, right? You know what I'm saying? And it's like they made it seem like it was this horribly complicated process, and it's not. And then you got people start throwing in there protoplast fusion and DDK and snake venom, and I want to punch those people in the face because I'm like, they're not. They are spreading misinformation, and they are basically trying to keep these secrets that they think are secrets. And my entire goal of life right now is to basically teach all of y'all those secrets, quote unquote, that they've been keeping, because I know all those secrets. and, and give me another 6 months, and y'all keep watching my YouTube videos, and I will tell you every single secret you ever need to know about growing cubes, mating cubes. To be fair, I still mess up. I still get tricked, man. Trichoderma. I still shit. I threw a tub away today that was all bacterial. It's like, yeah, whatever. Yeah, fuck it,
It
man.
happens.
Move on. Yeah, man. You gotta move on, or they get back up on that horse. Choir, whatever how people say it is. It's like it's not that expensive.
I actually threw
Move on.
I actually threw away a few tubs with Tretch, and I've only been doing it 8 months, and that's the first time in a while that I've had it. So it happens, I guess,
I mean, the
right?
seasons, man. When I first started, like back in college, like the seasonal changes, the humidity, the daylight, even. If you're growing on, in maybe your living room or in your bedroom, little subtle things like the temperature of the wall. Like I put my tents up against the internal wall, but the temperature of the wall and the floor, it's like a 2 degrees Celsius. It's about 5 or 6 degrees Fahrenheit. It's a difference. So if I've got my tents like right up against one wall, like that wall's like, I don't know, we use Celsius here. So say one wall's like 74 and the other wall's like 78. That can significantly affect the conditions in the tent. So like if you got trick, I mean just a random guess. If you're getting trick now, it's because the relative humidity has changed in the ambient environment, and that is going to, whether it's a monotub or a tent or a bag, that is going to affect the conditions inside the bag, right? It's going to affect transpiration rates, the overall growth, the temperature. And so the seasons, man, back in Michigan, probably, where you're at too, it's like I mean, you go from summer to winter, spring, fall, whatever, and the conditions are going to change dramatically. And you think like, oh man, this worked for 6 months and now it's all going to shit. Don't worry. Just wait another couple. Just keep going. That's the one thing. If there's one thing, do not give up. I failed miserably, and I have a friend now who's having a really hard time. And you just got to keep. That's why we need this community, because every time you got a monotub that you're throwing away and it's all tricked out, you got to be somebody's got to be out there to be like, don't be a like, pull yourself up, get back up on that horse, try a new cultigen, can order a new block of quar, whatever, man. Clean your monotub, get the bleach out, and buy a new bottle of alcohol, spray your body, whatever. Get drunk and forget about it. Do it again. Do it again the next day or the next week or the next month, because you know the rewards that you get from this hobby are not really, you can make a little scratch on the side, a little side hustle, but the real reward comes from this, and that's where the future is going. It's the actual cultivation process. You are growing something, right? You are growing something that normally doesn't grow in a fucking monotub, right? There's something literally magical about that. Like, you have to look at those things and be like, oh my God, look at what I've done. Like, I have just taken a fucking inert jar of water or whatever you started out with and like turned it into this living organism. And it's like, this is pretty damn cool. Right? Like this is pretty cool. Like, and I did it in like a month. You know, if you're growing plants, you know, I used to have gardens all the time. And man, you can grow tomatoes or weed or whatever. It takes 3 months, 4 months. Man, we could have a generation in a month and a half, right? So if you're into breeding, like that's the one great thing is if you want to find new stuff, which a lot of people do, you know, name it after your dog or your guinea pig or whatever. Like you can do it so much faster with fungus. Um, so yeah, we can get back into that cloning thing later if you want, but that's like kind of a more difficult thing to discuss verbally. Like it might be better to like write things down. If like I said, if there's people out there, man, just have them send me personal. What are they called? DMs now? Yeah, direct
Yeah, yeah,
messages. But
DMs, yeah.
yeah, you can DM me. I mean, like you know, if I might get overwhelmed if it's like 1,000 people or whatever, but I don't think there's that many people listening. But if you guys want
No,
to
no.
DM me
I
or
think so
whatever,
far
just
we've had what, 11? So
oh, okay,
it's still
okay, good. So they won't.
it's
So the mail bombs won't come yet.
I think it's more afterwards, like once it's been played, people start downloading it and the lot
Oh,
like
that's right.
yeah, they start downloading and listening to it and stuff like that.
Okay, so I'll be getting death threats over the next couple of weeks, not just tonight. It'll be like the next 2 weeks. It'll be like slowly trickling in. Like, Ed, you just gave away our secret DDK protocol. You shouldn't have done that. you should you shouldn't have gave away our monokaryon mating pattern. That's secret. That's like Illuminati information. You shouldn't have said that. Good. That's okay. If somebody, I invite them here and I can come visit. I'll take them on vacation, take them down to the beaches. They can see. I'll be their tour guide.
Does the fungi grow on the
Uh,
beaches down there?
now, now, maybe on your body, maybe on your human body after you've been, you know, out at the bar many, many nights and you're sun like immunocompromised. Maybe then you'll get fungus, but it won't be in good places. I can guarantee that. All those moist places on your body, yeah, you could probably get some fungus. But now they have a lot of mountains and stuff up north here. Like when I say up north, like it's like kind of up near like those mountain stuff. Like when you see all those pictures of like people wearing these funny kind of looking outfits and stuff. They live up in the mountains, and that's where you really got to go to find fungus because like Bangkok is pretty much a concrete jungle. You know, you might as well be in like San Francisco or New York or whatever, DC. It's just like concrete everywhere. So yeah, everybody lives in condos, apartments. There's not really a whole lot of nature around here.
Blue Michael wants to know if he can cut a piece of egg. Oh, he says, "Hope this isn't a tacky question about LC, but can you just cut a piece of agar and drop it in some LC broth versus using a needle?"
No, hell yeah, dude. That's exactly the way you do it. So I was sorry. I hoping I'm gonna use the bathroom. Sorry if
Okay, time
if
out.
you hear anything.
Time out.
Oh no, it's all good, man. I got the phone's down, but I don't know how good my microphone is, but yeah, I'm a human. Uh, but yeah, that's exactly what you do. If you want to make LC, the best way to do it is get a clean plate on agar and literally take a chunk of that and very, very carefully open the lid on the LC and just put it in there. What I like to do is if you're using a scalpel, cut a square out and just barely stab it on the end of your scalpel, and when you put it in the LC, kind of tap the blade, and so it'll just like fall in. Like don't stick it down. You just like want to tap it on the rim of the jar and kind of like let it fall down in, and then get the lid back on it as soon as you can. And if you do it in even like an SAB, like speed is not moving fast, but efficiency. I think they say like efficiency of movement. You don't want to like be making unnecessary movements. And so if you can you get your plate, you open it up, you cut your little square, and then with your other hand, you're already kind of reaching for your LC jar, and you're unscrewing it, and then you just tap it in there, and then you get it on top, back on it, and then That's it. It's really pretty straightforward. Like when you're making LC, I think I got a video about that too. I'm
Yeah,
pretty sure I do actually. I got
I
a
think
video
you
about
do.
that. So there's a that's yeah, LC really easy. It's easy to get contaminated, but once you get it down, it's by far. I just discovered there's a book called Radical Mycology. If anybody is looking for a read, it's dense though, man. It's really dense. It's a big book and it's pretty technical, I guess, as far as our hobby goes, but it's one of those things like you can read a page and you read that same page like 20 times and you'll still be finding little bits of information like, wow, that dude Peter McCoy, I think is his name, he is like clearly a very smart person but also well read. And you don't see him in kind of the cube community, but the techniques he's talking about are clearly oriented towards any kind of fungus. So he's not just like Paul Stamets. Paul Stamets never really talked about cubes, but it's obvious that like those techniques can be applied to our little babies.
I actually,
But yeah, LC's dead easy. Once you get it down, I was actually running everything off LC for a while, and you can propagate, and if you need a little need a little extra money for rent, man, you sell LC. Man, you can make a liter of LC for like 50 cents, like a quarter. Like I don't know, you guys, yeah, like a quarter US. You make like 20 LC syringes. Like here, the syringes cost like about a nickel, the needles like a penny, and they're all sterile. And like you sell them for 20 bucks. The math works out to your advantage in that case. So yeah, that's like what a lot of people are getting into that because it's really dead easy to do LC. And you can when you make 10-20 bucks for a freaking syringe that costs you like a quarter, it's a pretty high profit margin.
yeah,
So, and it's also
I've just been giving mine away. So
yeah, me too, the same here. That's like I was saying, the only reason I ever thought about charge, I just got to go to the post office, a pain in the butt. But if people do order from that Spore Swaps, man, I usually like somebody orders like a $15 swab, I throw in like two extras or like something like yeah, like same card, same thing. I'll just like yeah, there's a Choco Tat and a Chota Mock and like whatever else is laying around.
I'm gonna have to hit you up.
Because I just
I'm gonna have to hit you up for some spores, man. I need some of your cultures.
yeah, well that's the thing. I mean, if you DM me and you like talk nice, like maybe I'll send them for free. I mean, I'm not trying to like have 1,000 people DM me for spores, but if you kind of chit chat a little bit, and maybe we can trade stuff, like whatever. Like like I said, I'm not in this to make money.
No, I'll pay you. I'll pay you for your delivery and stuff like that, man. I'm gonna have to hit you up.
Yeah, yeah, that'd be awesome. I mean, that's it, and then it's easier because they got like a website and it just sends me an email with all the stuff like your mailing address and all that shit labeled and everything.
And
It's
it's
it's,
called
it's a lot easier to
Spore Swaps.
uh swaps. Yeah, it's for swaps.com. I think it is .com. They got like a Facebook thing, but they switched over to some website because they were having trouble with Facebook, of course, banning pictures and all that kind of stuff. So yeah, I think it's just Spore Swaps with an S at the end.
Yeah.
So yeah, I'm sure like I say, if somebody DMs me or whatever, I'd rather like it's easier just if you got 15 bucks. The problem for me here is like it's hard to do like international money transfers and all that. Like so if somebody wanted to just like give me $5 for like shipping, it's hard. It's like impossible basically for me to do that here. I've got to do it all through like my friend back in the U.S. and it's just a pain in the ass to be honest. But so it just makes the 15 bucks like worth all the pain in the butt. But like I said, man, I'm pretty generous, man. When I'm packing up that little greeting card, like I tend to put extra. Well, I don't tend to. I do always put extras in there,
Oh,
because sometimes.
I actually have that site on the Discord here. It's made by Sherry Berry.
Yeah, yeah,
Yeah, I
exactly,
think she was in
exactly.
the
That's cool.
Discord here for a while, but I think she left.
Yeah, she's quite busy, man. She's got to deal with all kinds of people. Like, yeah, she's a busy woman. I think as a woman, I'm assuming it's a
That
woman.
sounds like it.
Um, yeah, she's like quite busy. Like so, yeah. If she don't, yeah, she's got to. I mean, she's got to deal with a bunch of people like us. Like it's like, yeah, it's a hard job, I imagine. You know, some of those people that have short term memory loss, like 24 hours a day.
Yeah, that's me.
you know, those kind of people, yeah. Yeah, I know, man. I know some of my friends have become like professional, well, smokers. It's like, wow, because it's like every weeds everywhere here now. So, like, you got like this whole generation of like people that are like, now I'm just going to smoke weed all day, every day. Like, it's not that many, but I mean, I smoke too, but it's like not something I do 24 hours a day. A lot of my friends do. So, yeah.
I'm looking on this site here, and it says they have like a plate or whatever, and it says research plate. What is that? Like, what is a research plate?
Yeah, that's a fancy word for them basically selling dykarons on agar. Hopefully the FBI or whatever's not the DA is not listening. That's the new. So like they used to call syringes tubes. That was a fancy name for basically a spore syringe. And then tube kind of turned into what was basically an LC, like a liquid culture syringe. And so now they're calling, I believe, research syringes are LC.
Uh,
And I don't even. And so a research plate is essentially going to be a dikaryon that is already plated. So if you want to get an actual plate that you could use to make spawn like immediately, assuming it's not all like old or fucked up or whatever. You could use that spawn and go A2G agar to grain. You could just immediately go to grain. Or if you were smart, and sorry for those people selling, if you're smart, you'll make LC, and as soon as you get that plate, you'll do two things. You'll sub it to a new agar plate, and you'll make LC out of it.
huh.
So this is back to the reason why some of these people who are selling stuff have to charge like an up. It's almost like a one-time price because they know once you get that plate, if you know what you're doing, you're never ever ever gonna buy from them again. See what I'm saying? Like one of the fascinating things about what we do is that we can basically get a culture and have that culture forever, assuming it doesn't do any weird stuff like revert or whatever. Not only that, once you get spore prints or a swab, you can basically store those for years and years and years. And if you learn how to do long term storage, like the water vials and slants, like you essentially become self sufficient. So if the cordyceps, do take over the humans and, the last of us or whatever that show is, if that does happen, like it doesn't matter. We'll still have plenty of things to do because we'll be down in the bunker, like growing cubes and bartering with people for ammunition or whatever clips, like we do. Like if you have like the things we can like grow underground in the dark with no electricity in a plastic container, like you're pretty much set. Like you don't have to worry about the zombie apocalypse because you'll always have something to barter with.
That's like what was it? Oh, P Funk. He sent me a illustrated AI picture, and it was like this mad scientist guy, and he was just like exactly like you were just saying. It looked like he was down in the basement with his beaker, and it's just when you're saying that, that picture came to mind.
Yeah, exactly. Oh, I think yeah, I meant somebody mentioned that on another pod. If you learn how to make your own alcohol and grow mushrooms, like you will always have something to barter with. Like in the post-apocalyptic world, people are gonna want to do two things: get drunk and like forget about reality. And once you learn how to, wine's dead easy too, man. We could do a whole other podcast about wine if you want, but that's easy to make. But yeah, yeah, I don't know if you had a time limit, 2 hours,
Yeah, there's no time limit, but
or do you have any more questions? I'm curious if somebody gave you a question, it might be nice to get to it so
hold
that
on.
we
I
don't
think
have to hang in or
there
anything.
we. Oh,
There might be a bunch of questions in there now. Like I don't know. I'm not. I might check on the Discord thing there.
what's
I had to restart my computer. My computer started asking me that question about like, oh, do you want to update and blah, blah, blah.
um? I don't. Well, hold on. Would it be through?
It's quite funny.
Oh.
I can smell my neighbors must be smoking weed. I can like smell it. This is like Saturday night here. It's like midnight Saturday. It's like, yeah, I can smell weed. Somebody's smoking weed.
How does it smell?
What?
Like a skunk?
It smells really. It smells pretty damn good. Or oh wait, it could be. No wait, my bad. I just sat down at my desk. It's my weed. Yeah, that. It's my. I forgot. I completely forgot. Yeah, it's my Gorilla Glue. And what is this? Studio 59. Yep, that's it. They got weird names. They got like 1,000 different names now. But brain damage, red poison, gelato, sour diesel, cherry pie, banana kush, blah blah blah.
But it's all the
So
same,
many
right?
of them now. Oh yeah, that's another the cannabis. Now they've literally started. They used to say like, oh, it's 60% indica, 40%. No, you would see. I asked them. You'd see signs that were like, oh, it's 60% indica and like 30% sativa, and I'd be like, well, what's the other 10%? And they're like, I don't know. And I'm like Ruderalis? Is
Hairspray.
it like, or is it? Yeah, that's it. The other, it's like the petrol, the gasoline, hairspray, like embalming fluid, formaldehyde. Like, what are they putting on it now? PC, PCP or like what? Like ketamine, fentanyl. I don't know. What are they putting on it now? Well, that's another thing. Speaking of that, man, they're starting to put fentanyl in even like the shakes here now. So people will go to the beach and they'll get like a mushroom, and one of my friends was like, "Oh yeah, we were at the beach and I got a shake, and it should have like maybe 5 grams in it max, 3 grams probably, maybe 2 or 3 is gonna be standard, dried." And this guy was like, "Oh yeah, man, after like 10 minutes, I was so fucked up," and I'm like, "Oh dude, that's not fungus. Like you don't get fucked up that fast." And then he described the rest of the night to me, and he's like, "Oh yeah," and then it's like, dude, you got dosed with like fentanyl, like mushroom. Even if you're doing lemon tech or whatever, man, you're not gonna get that fucked up after 10 minutes. And I was like, wow, they are now dosing people with fentanyl. Over here we're a lot closer to China, so like people go online and buy a key of it, and it's a fucking worldwide epidemic, man. It's kind of scary to be honest. But so that's I don't know, going back to the zombie apocalypse thing. When you can make your own medicine or your own entertainment or whatever you want to call it, like it's a good thing. It's a very very good thing. Oh god, now my YouTube's coming on.
All right. Well, I don't know. I don't think I have any more questions, and I don't see any more. You got anything else you'd like to talk
Yeah.
about?
Not really right now because I reckon, yeah, like I said, if you want, I can be on like whatever, however, if you want me on
Of
again.
course I do. Of
But
course, you don't have to ask that.
I would, I'd love to go into like more topics in like more detail. I kind of want to get a feel for people's knowledge level. If I'm talking, I don't want to talk down or above people. Like kind of want to get a good like gauge of like what people are interested in and like how deep they would want to go into certain things. Like I could go into excruciating detail about maybe some of the the more the genetic stuff, but I don't know if people are really that interested in it, to be
I
honest. Like I
think
don't want to bore.
I think they'd enjoy that. Like we can set up a certain podcast for, like one topic at a time if you want, and
Oh,
kind of make it like a online book kind of thing, or like I said,
yeah,
most
that
of it's downloaded. So when people see the titles or like stuff like that, they download it or they move on. You know what I mean? So we can
yeah, that's a good idea. That's a is it like
kind of like an online book, like like certain topics, online book kind of thing. That's what.
yeah, that was sort of the idea with me starting the YouTube videos. The only thing about like that, I kind of do them sporadically, so there's not really much of an order to them. Like so one of these days I'll have to go and make like a play. Is that like a playlist for the
Yeah, you got to
videos?
put them in a playlist and organize it all, and
That means I'll have to go watch my own videos. Like I don't want to watch my own
you got
videos.
to label them. You got to label them. You got them labeled, like you know.
I do. The problem is some of them they're like they're like multiple topics in the same video, so it's like oh
Well, it's like
like ow like I was talking about monos, but now I'm going off about swabs, and now I'm talking about liquid culture, and like it's like
the mushrooms. Then
which
throw them, clean them up, start fresh, and stay on topic.
no
Write a script.
I'll nah
Too
nah I'm
much
never
work,
gonna
eh?
write a script. Oh, it's way too much. I'm never when I started doing those YouTube videos, two things which I shouldn't say. I was like, I'm never gonna write a script. I might write like bullet points, but I'm never gonna write a script, and I'm never gonna edit it. I get a little bit annoyed with these hyper edited videos where they like maybe did something that you should have seen, like a mistake that they should have showed you,